Hi everyone, my name is Fatemeh Gholizadeh, PhD student from Concordia University. The title of my study is the modulatory effect of nicotinic acetylcholine receptors on human memory T helper 1 and T helper 17. At the beginning, I would like to have a general introduction on acetylcholine as a neurotransmitter in parasympathetic nervous system. Acetylcholine can target skeletal muscle, brain, and many other organs, and following that, acetylcholine can regulate some functions in the body like heart rate, respiration, and so on. In order to do its job, acetylcholine to do its job, it has to bind to some receptors on the surface of its target cells. So those receptors are called muscarinic receptors and nicotinic receptors. Recently, it has been proved that immune cells can have their own cholinergic system, means that immune cells can produce their own acetylcholine and their own receptors, like nicotinic receptors. Speaking of the immune cells, adaptive immune system involves two parts, cell-mediated immunity and humoral-mediated immunity. Naive T helper cells and memory T helper cells are two populations of the cells in the cell-mediated immunity. Regarding the naive T helper cells, when antigen-presenting cells present antigen to naive T helper cells, depending on the cytokines in the environment of the naive T helper cells, they will differentiate into different T helper subsets. In this study, in our study, the main focus are T helper 1 and T helper 17 cells. T helper 1 cells produce mainly interferon gamma, and T helper 17 secrete a lot of IL-17. Memory T helper cells are the other part of cell-mediated immunity. There are different hypotheses about the formation of memory T helper cells, but one of them proposed that when antigen is cleared from the body, most of the effector T cells will undergo apoptosis. However, some part of the cells will form memory T helper cells. Memory T helper cells in our body, they will reside in the tissue or they will circulate in our body, and in the case of re-exposure to the antigen, they're gonna secrete the cytokines more robust. It has been proved that individuals diagnosed with conditions like MS exhibit higher level of memory T helper cells. So it was the reason that in this study we chose memory T helper cells to work on. This slide is showing the combination of the cholinergic system and immune system. As you see here, T helper cells can produce their own acetylcholine, and also they have their own nicotinic receptors. Nicotinic receptors are ligand-gated ion channels. They are pentamers, and these pentamers composed of 1 to 5 different subunits. One of the subtypes of nicotinic acetylcholine receptors are alpha-7 nicotinic receptor, meaning that all of those subunits that are forming receptors are alpha-7. Eventually, acetylcholine binds to the receptor. The conformational changes lead to this receptor to be opened, and some of the ions like calcium and sodium move across the receptor and change and depolarize the target cells like T helper cells, and following that, some of the signaling pathway will be activated. This slide shows that when acetylcholine binds to these receptors, nicotinic receptors, some signaling pathway will be activated like phosphorylation of JAK2, and it would interfere with the phosphorylation of the I-kappa B, so it would prevent the recruitment of NF-kappa B and consequently suppress NF-kappa B dependent pro-inflammatory factors expression like IL-17 and interferon gamma as the main cytokines produced by T helper 1 and T helper 17. Previous studies have shown that nicotine lead to an increase in IL-4 and decrease in IL-17A in T helper cells extracted from rheumatoid arthritis patients. Or some other studies have shown that alpha-7 knockout mice, in the alpha-7 knockout mice, nicotine could diminish the production of pro-inflammatory cytokines by alpha-7 mediated pathway. Previous studies suggest that nicotinic acetylcholine receptors are involved in regulatory immune function, but the role of nicotinic receptors, particularly alpha-7 receptors, in nuclear signaling and regulation of cytokine production in memory T helper 1 and 17 cells is still unknown. So in this study, we investigated the involvement of nicotinic acetylcholine receptors in the regulation of memory T helper cells and cytokine production. To reach our goal, we had to design our study. So for the material and method, we obtained PBMCs from healthy participants, and then we isolated memory T helper cells, and we did cell culture with the nicotine and GTS21. Nicotine is an agonist for all of nicotinic acetylcholine receptors. It can mimic the effect of acetylcholine, and GTS21 is an agonist for alpha-7 nicotinic receptors, and it binds to just alpha-7 nicotinic receptors. And after 5 days of incubation, we ran ELISA, flow cytometry, and Western blot to measure cytokine levels and some proteins. Regarding the results, we started with PBMCs. We exposed PBMCs with the nicotine, and we figure out nicotine could significantly decrease the level of interferon gamma. In all of my results section, ACT is a condition that the cells receive anti-CD3 and CD28. So for interferon gamma, the level of interferon gamma significantly decreased after treatment with nicotine, but for the IL-17, there was no significant changes, but the result of interferon gamma was a hint for us to work on more purified cells like memory T helper cells. And surprisingly, in the memory T helper cells, the level of interferon gamma and IL-17 significantly decreased after treatment with nicotine. In order to confirm the ELISA result, we did intracellular staining to measure the cytokine levels at the cellular level. We figure out that after treatment of the cells with nicotine, the percentage of CD4 positive, IL-17 positive significantly decreased. For the interferon gamma, it was the same. The percentage of CD4 positive interferon gamma positive T cells after treating the cells with nicotine significantly decreased. This result showed that nicotine can suppress memory T helper 1 and memory T helper 2. To see which signaling pathway is involved, we measure phospho-NF-kappa B per NF-kappa B ratio. So we expose memory T helper cells for 15 minutes to the nicotine, and then we measured phospho-NF-kappa B level. We expected the level of phospho-NF-kappa B, or this ratio, if we convert the results of ACT and ACT plus nicotine, we expected a significant decrease here. But the results showed that we had just a decreased trend. So maybe some other signaling pathway are activated, and we have to measure some other transcription factors and study other signaling pathway in this case. And then, in order to see what would be the impact of the nicotine on the proliferation of the memory T helper cells, we did CFS staining, and our result showed that nicotine could not change the proliferation. As a division index and the proliferation index, there was no significant differences between ACT and nicotine group. Moving forward, we thought it's better to target a particular nicotinic receptor subtypes because nicotine usually binds to most of the nicotinic receptors. So we decided to work on alpha-7 nicotinic receptor because recently they've drawn a lot of attention in different cell types. So we use an agonist called GTS21, which is solved in DMSO. So vehicle control in this case is DMSO. So GTS21 as an agonist binds to alpha-7 receptors, and the ELISA results show that after incubation of memory T helper cell with the GTS21, the level of interferon gamma significantly decreased, and the level of IL-17 also after 5 days of incubation significantly decreased. So those results showed that GTS21, while alpha-7 nicotinic receptors is suppressing the interferon gamma and IL-17 production. In conclusion, activation of the nicotinic receptors, particularly alpha-7 nicotinic receptors, suppress memory T helper 1 and T helper 17 cells, and our findings suggest that targeting nicotinic acetylcholine receptors in memory T helper cells can be an approach for manipulating immune responses to control the inflammation in autoimmune diseases like MS. Since my project is an ongoing project, our future plan is to knock out alpha-7 nicotinic receptors gene, which is named CHRNA7, because we want to see when we have loss of function of the alpha-7 receptors, what would happen. We want to make sure the visualized effect is mediated with alpha-7 nicotinic acetylcholine receptors. Also, we would measure alpha-7 receptors, STAT3 and FAT1 and FAT2 and some other transcription factors and signaling pathway at the mRNA and protein level to figure out which signaling pathway is activated in the downstream of the nicotinic receptors and which signaling pathway lead to the suppression of memory 1, TLP1 and memory TLP17. And finally, we would like to work on memory T helper cells in the EAE mice model because we want to see how is the effect of alpha-7 nicotinic receptors in the pathological conditions like MS. Finally, I'd like to acknowledge my supervisor, Dr. Darlington, and all of the lab members because of their help and support. And of course BSI because of giving me this opportunity to present my work. And thank the audience for the attention.